Okay, welcome back. Week 8, we're looking at the skeletal and muscular systems. So this is chapter 7 and chapters 8. We kind of group these two systems together because when we talk about movement, it's actually a combination of these two body systems working together. In order to move your body, you have to apply tension to the bones, and the tension is applied by the muscles that are attached to bones. We call them skeletal muscles. So movement is happening as a result of a cooperation between these two body systems. Skeletal muscles are the muscles that are all over the body, right? We go to the gym, we work those muscles out. And as they contract, what happens is that they will pull on the bones, and as a result, movement is what happens. So the muscles never push on bones. There's only when muscles contract, they become shorter. And so the end result is always a pulling on a bone. And that's how we move around. But in addition to that, there are some other functions of the skeletal and muscular systems. Besides providing movement, there are bones scattered around the body that are there to protect vital organs. The cranium is one of them. The rib cage is another, protecting the heart and the lungs, including the sternum. The vertebral column, of course, protects the very delicate spinal cord. Your muscles are involved in generating heat. Remember, when your body temperature drops a little bit, one of the results would be to shiver. And shivering is a rapid contraction of skeletal muscles, and that's going to generate heat. Our skeleton, along with our muscular system, allows us to maintain that upright posture, along with bipedalism. That is a very unique characteristic for humans. It helps us to manipulate our environment. And in addition to that, if we look inside the bones, specifically in the red bone marrow, this is where we're going to be producing our blood cells. And this is where we could also store minerals and other materials. Like in the yellow bone marrow, we store lipids, fats, but also in the matrix of the bones, we could store some minerals, calcium and phosphorus, for example. All right, so movement, again, is possible because of the unique arrangement of muscles being attached to bones. Muscles attach to bones through these connective tissues called tendons. And as the muscles are contracting, they're getting shorter, right? And as they become shorter, the result is to pull on the bones. And then, of course, when we relax the muscles, they go back to their original length. Sometimes we could stretch or elongate, right? If I have like this elbow joint here, is called a hinge joint, right? When I contract the biceps muscle, it closes the joint. When I contract the triceps muscle, what happens is it relaxes the bicep muscle and it opens the joint. I could elongate the bicep muscle if I try to hyperextend the joint, which is possible, right? We stretch our muscles sometimes. Okay, so we're looking at bones and muscles, and these are classified as connective tissues. They're made of cells just like the other tissues in the body. Therefore, they're living tissues, right? We can't have movement with only one without the other, right? We need both. Okay, so remember that the muscles pull on the bones only. They never push on a bone. At the joints, this is where two bones meet, and this is where movement is going to be possible. Not all joints are movable joints. However, some joints are immovable, like the joints in the skull. However, the joints that are scattered around, like in the shoulders, we have ball and socket joints, and fingers, and in the elbows and knees, we have hinge joints that allow for movement. In lab, we talked about the difference between compact bone and spongy bone. Compact bone is more dense. We find it closer to the edges of the long bones. And when we look at it under the microscope, we can see that the arrangements of cells are, they take the form of circles and rings. And this network of rings that we find making up compact bone, these are called osteons. So the osteons are the functional units inside the compact bone. So we have concentric rings of bone matrix. These bone matrix have been laid by specialized bone producing cells called osteoblasts. So what the osteoblasts are doing is they're releasing a substance, and that substance is kind of like a gluey type substance. It's called osteoid. And inside the osteoid, there is a collagen that has calcium binding proteins in it. And what happens is as the calcium adheres to this osteoid substance, it becomes more and more solid, right? And as a result, what happens is the osteoblasts actually get trapped inside of the matrix. And as they mature, they go from being osteoblasts to being more of a mature version of these cells, which we would refer to then as osteocytes. So after we're looking at them, right, the images that we saw in the lab, we saw the Haversian systems or the osteons, and we saw the osteocytes in lacuna. So it's at that point that the matrix is already formed. The matrix has become calcified, so it's hardened. And now those cells become entombed in the lacuna, right? And their job is now shifts from a bone building situation to more of a bone maintaining situation. So the osteoblasts are the cells, the immature cells that are actually producing the bone, and then they mature into osteocytes. And their job is more to maintain the bone. All right. Spongy bone, on the other hand, we see that more in the epiphysis of the long bones, in the deeper portions of the bone, not so much closer to the surface where we find the dense compact bone. Spongy bone is more porous. It's also referred to as trabecular bone. And that spongy-like appearance, we refer to that as the trabeculae. Right? We don't see osteons there. We see trabeculae or struts. So if you look at this image over here, towards the surface of the bone, this is where we see the dense compact bone. This is where we see osteons. And you can see the osteocytes arrange themselves in circles. Every black dot in this picture here is an osteocyte. And there's this concentric ring system that we see around the central canal. It's inside the central canal that we would have blood vessels and nerves. And these cells arrange themselves in circles around that central canal. So this matrix here is calcified. It's hard and it's very dense. If we look over here at the trabecular bone, this is spongy bone. We don't see osteons over here. It's more of a porous bone. And what's good about spongy bone is it offers strength. This arrangement it offers strength, but at the same time, it's not as dense, so it offers lightweightedness. If our entire bone was compact bone, our bones would be much more heavy than they are. So this is a nice design that allows the bones to still be strong, but yet keeps them as lightweight as possible. So it's in the central canal. Again, we have the blood supply, we have arteries and veins in here, and there are nerves in there as well. Okay, so the osteocytes, as they become kind of trapped in this calcified matrix, well, they survive inside this little chamber called the lacuna. Lacuna actually means tiny lakes, but what are they? They're just small holes that the osteocytes live in. So how do the osteocytes all the way out here on the outer edge get the nutrients and oxygen from this blood vessel here? Well, you'll notice that from the central canal extending outward to the lacuna are these little lines. These are tiny canals, and these are called the canaliculi. We could see some definite canaliculi extending from this lacuna with an osteocyte. You can see the lines going from that one to this one, and then from that one to this one. The ones in the front row have lines that go directly to the central canal. So materials that are passing by through these blood vessels get exchanged even to the cells that are far out here through these tiny canals called the canaliculi. And that's what keeps these cells alive. Again, when we look at the long bones, we have the ends of the long bones. We refer to that as the epiphysis. And then the shaft of the long bone, we would call that the diaphysis. You could see at the edge of the shaft, this is where we find compact bone. But the compact bone isn't confined to the diaphysis. The compact bone actually will go into the epiphysis as well, and we find it very close to the surface. Deep to that, we see this trabecular bone, or this spongy bone. All right. At the ends of the bones where two bones meet, we would see articular cartilage, right? Because this is where, let's say, for example, a joint would be here. So there would be rubbing of two bones together. So to minimize damage, we have this protective layer of articular cartilage. We also see in the epiphysis this structure called the epiphyseal line. And this is the last portion to be converted from cartilage to bone. And this happens after growth is complete. So if you see a bone and this epiphyseal line is soft and cartilaginous, then you would know that this bone is from an immature organism. However, if you open a bone and you see that this has become calcified, well, then you would know that that organism has been reached maturity and is finished growing. All right. In the epiphysis that we see in the spongy bone, this is where you're going to find red bone marrow. All right. And it's in the red bone marrow that the blood cells are going to be produced. So we have hematopoiesis. Poiesis means the production of hematocytes, right? So blood cells. In the diaphysis, this is where we find the medullary cavity. And what fills the medullary cavity is yellow marrow. And yellow marrow has a high concentration of fat. Remember, we store fat as a long-term energy source. So there's a couple different ways at which we can produce bone. This picture is showing us what's called endochondral ossification. Endo means within, chondral means cartilage. So we're producing bone from inside the cartilage. Now, remember, when we are forming our bodies, right, as embryos, right, our skeleton begins as hyaline cartilage. And let's say, for example, this is one of the long bones. So as the femur, for example, is being produced or being developed as an embryo, it will start to take on a shape. You'll start to see an epiphysis at one end in the diaphysis, but it's completely uncalcified. It's completely 100% cartilage, and it will grow like that. Eventually, what's going to happen is we're going to have an artery showing up. And once an artery shows up, it's going to generate what's called a primary ossification center. All right, and here we see this happening in this picture. This is an artery. It has become attached to the diaphysis, and it is becoming what's called a primary ossification center. So what's happening is the osteoblasts are being laid down, and they're secreting osteoid substance, which is going to convert this hyaline cartilage into a calcified matrix. So it's going to convert the cartilage into bone. And this is going to continue until it takes up the entire diaphysis. So the diaphysis is completely bone. Now, we're still going to have a large percentage of the epiphysis that is cartilage. So what's going to happen is a secondary ossification center will be formed, and that's going to happen when blood vessels appear in that area. And then osteoblasts will appear there, and then again, converting that hyaline cartilage into bone. So this is a secondary ossification center. So osteoblasts will appear, osteoid substance will be laid down, and then that matrix will become calcified, turning it into bone. So what's left? Well, a little bit of cartilage on the tops, which is what we like, right? Because that's our articular cartilage. That's going to protect the bones. And a little bit of cartilage in between the primary and secondary ossification centers. And that's what's going to make up the epiphyseal line. So as the bones are being produced, or as the bones are growing, rather, they're going to be growing from this line, producing more bone at that line. And that's what's going to happen then is the bone is going to elongate until you reach maturity. And then that begins. And it's this line that is going to be the last thing to become ossified. All right, so growth occurs at the outer surface of the bone also. Now, this is a different type of ossification. What we just looked at was called endochondral ossification. This is more of intramembranous ossification. Intramembranous means, intra means within, membranous means the membrane, right? And there is a thin membrane that covers the bones. It's called the periosteum. Peri means outside. Anything oste or osteo is bone, right? Peri, like for example, we see the word perimeter. That means outside, right? You measure the outside of your property. That's a perimeter. So peri, a lot of times, is a prefix that is used to mean outside. So periosteum is outside of the bone. There's a very thin membrane, and it's filled with osteoblasts. So what can happen is, unlike the primary ossification center establishing itself deep in the hyaline cartilage model, now we're seeing osteoblasts positioning themselves on the exterior of the bone, and when they start to produce osteoid and produce bone, it causes more of a thickening effect, right? A thickening. So this is intramembranous ossification is involved more in the thickening of bones, where endochondral ossification is involved more in the elongation of the bone. All right, again, but as osteoblasts are releasing osteoid and the matrix becomes calcified, these cells become trapped inside the matrix and they mature into osteocytes. And it's the job of the osteocytes to maintain the structure of the bone. There's another type of bone cell besides osteoblasts, which will mature into osteocytes. There's also osteoclasts. Osteoclasts become active when we need to remodel the bones from the original bone formation. And this happens more often than you would think. Your bones are constantly being remodeled. Remember, one of the functions of the skeletal system is to store minerals. We store calcium and phosphorus in our bones. And if the blood is low on those minerals, then we need to release the calcium from the bones. And so it will be allowed to enter back into the bloodstream. We release calcium by using bone dissolving cells, and these are called osteoclasts. So when osteoclasts become activated, they break down the calcified matrix, and that releases the calcium into the blood from the bone. When blood calcium levels are high, then the osteoblast would take the calcium from the blood and add it to the osteoid substance and store it back into the bones. So when it comes to remodeling bones, for example, if you have like a radical change in lifestyle, where you decide that you're going to start to go to the gym every day, where before you never went to the gym, and you do a lot of leg exercises and you're putting a lot more pressure on your leg bones than normal, the bones will slowly begin to remodel themselves in order to better handle this new type of activity. And what they can do is they can take bone from one side of the bone and dissolve it and then add to another side of the bone. Because as we don't use our bones, they could take on like a different shape. And as you use the bones more and more often, they will remodel the shape, right? So they'll become curved if you don't use them as often as you should. And when they curve like that, they lose their structural integrity. So remodeling of bones is a very common thing, and it happens as a result of the interplay between osteoclasts dissolving bone at one point and osteoblast rebuilding at the other end. For bones to heal, obviously, if you break a bone, there's a couple different possibilities for this fracture to become aligned and immobilized. There's one type is called a closed reduction, and this is when you would go to the doctor and the doctor would just feel the broken bones from the outside of the skin, and without opening up the skin at all, just kind of align the position of the bones back to where it's supposed to be, and then wrapping it up with a cast. However, when bones are broken really badly, the skin has to be cut open, and sometimes screws and plates and pins have to be used to fix the bone back into position. When that happens, this is called an open reduction. Now, here's the thing about bone healing. We do not want the bone to be completely immobilized, because that will slow the healing process. Because remember, when we use our bones more, it will initiate, I guess, no, not initiate. It will cause the bones to become remodeled, right? So when we put pressure on our bones, the tendency is the bone is going to be remodeled. It's going to want to increase the strength. So when you break a bone, you should be putting partial weight-bearing activities, and that's going to stimulate these bone cells to start healing the bones. When it comes to the anatomy of the skeleton, this is good. These slides are in your notes and they can become helpful to you, but this is just a review of everything that we did in lab. So again, we talked about the axial skeleton. We talked about the appendicular skeleton, including the girdles. We did this in lab. I'm not going to do it again here, but this is anatomy. Anatomy is more for lab. This is another good picture of the vertebral column. The first 7 are the cervical vertebrae. The next 12 are the thoracic vertebrae. The next 5 are the lumbar. And then we have 5 fused, making up the sacrum, and then the coxal bones. And the ribs again, the first 7 are true because the articular cartilage, this costal cartilage, articulates directly with the sternum. 8, 9, and 10 are called false ribs because the cartilage, the costal cartilage, piggybacks off the 7th. It indirectly articulates with the sternum. And then we have these two out here, 11 and 12. Those are called floating ribs. Again, the pectoral girdle consists of the clavicle and scapula, and then the upper limbs, humerus, radius, and ulna. The radius follows the thumb. Carpals in the wrist, metacarpals in the hand, and then 14 phalanges. Again, this is all in the lab, so I'm going to skip over all this. We can classify joints as either immovable, synarthrotic, immovable, like in the skull. These joints come together and they form sutures, right? Slightly movable. These are called amphiarthrotic. This would be like in the carpals and the wrist. There's a little bit of movement, more of a sliding movement. And then freely movable joints are called diarthrotic or synovial joints. And we find those where the bones are moving a lot. So in the knees, elbows, shoulders, ball and socket joints, hinge joints are good examples. These are called synovial joints because there are synovial membranes associated with them. And within the synovial membranes, there are lubricating fluids called synovial fluid. So we have a synovial membrane. Well, we have the bones. We have two bones. Then we have articular cartilage. But we have an extra layer of protection in the synovial membranes filled with synovial fluids, consuming this space, which is called the synovial or joint cavity. These are just, this is just a map of some more anatomical positions. When it comes to muscles and bones, when we close a joint, like at the elbow, when you flex your bicep and you close the elbow joint, that's called flexion. When you relax the biceps and contract the triceps and open the joint, it's called extension. When you stretch it a little bit beyond, that's called hyperextension. When you have your foot and you are raising your foot up towards your knee, that's called dorsiflexion. And when you lower your toes, like if you're going to stand on your tippy toes, that's called plantar flexion. Whenever you move the limbs towards the midline of your body, that's called adduction. When you move your limbs away from the midline, that's called abduction. There are three types of muscular tissue in the body. We have skeletal muscles. These are the muscles that we find attached to our bones. These are the only types of muscle that we have control over. There's also cardiac muscle. This is your heart muscle. We do not have control of that muscle, nor would we want to have control of the muscle. There's smooth muscles too, and these are the muscles that we typically find lining blood vessels and the digestive organs. Again, we don't have voluntary control of these muscles. So peristalsis, remember peristalsis is how we push food through the GI tract. That's all controlled by smooth muscles, more of a slow moving muscle, controlled by the brainstem or the autonomic nervous system. When we look at skeletal muscle, the function of the skeletal muscle is to move the bones. So we're moving the bones, so we're moving the body. If we look at the skeletal muscle, we can kind of break it down into the belly of the muscle, which is the middle portion of the muscle, the origin, which is where it attaches, and the insertion is where the other end attaches. So how do we tell the difference between the origin and the insertion? Well, the origin is usually the one that is closer to the midline. Or another way we figure it out is there's less movement at the origin. The insertion is usually further away from the midline. Or it is the area, it's the end of the muscle that moves more during contraction. All right. Muscle cells are called muscle fibers, and when they are being used, they shorten, right? And that creates tension that pulls on the bones. Here we see in the arm, this is the right arm, we see the biceps muscle here, and then we see the brachioradialis. On the other side, we see triceps. This would be the belly of the muscle. This is the belly. So the biceps would have two heads, two bellies, and the triceps would have three heads. Remember, sep comes from the root word cephalic, which means heads, right? Heads. The cephalic region is opposite of the caudal region. The caudal region is the tail. Cephalic region is the head. So anything cephalic or sep, that means heads. These muscles have little heads or bellies associated with them. The biceps, that means two heads. Triceps means this muscle has three little heads. Quadriceps, four heads. The muscles attach to bones through tendons. And we see antagonistic muscle pairings when we compare the biceps muscle that closes the elbow joint and the tricep muscle that opens the elbow joint. They are working opposite from each other. Therefore, they are an antagonistic muscle pair. The biceps brachii and the brachioradialis, they can both be used together to achieve the same movement. So closing the elbow joint, when you are at the gym and you have a weight in your hand and you're trying to exercise the biceps, you're actually recruiting neighboring muscles to help with that movement, and the brachioradialis is one of them. So these two muscles are working together to achieve this flexion. Therefore, because they're working together, we would refer to those as synergistic muscle pairs. This is just a list of the muscles. Again, this is an anatomy slide. It's there for your review. We're not going to do anatomy in the lecture portion because it's just going to be a repeat of our lab stuff. So refer to the lab videos for that. This is a great picture so we can see the bone, and attaching this muscle to the bone is a tendon. We could see the muscle fibers arrange themselves in these bundles called fascicles. However, when we pull the muscle fibers out, we could see these long skeletal muscular cells. And these skeletal muscle cells are made up of these individual contractile units called sarcomeres. And of course, there's going to be some packs of mitochondria in there because muscles require a lot of energy. So the sarcomere is the functional unit of the skeletal muscle cell. This is a contractile unit. It's made up of alternating proteins, both thick and thin proteins or protein filaments. The thin filaments are called actin filaments, and the thicker ones are called myosin filaments. When the myosin filament, which is the thick one here, you can see it has these little heads that kind of stick out. When the thick myosin filaments are allowed to latch on to the thin actin filaments, what happens is they latch on and they pull in, and this whole sarcomere becomes shortened. So how do we allow this thick myosin filament to adhere to these thin actin filaments? Well, we have to release calcium because there is something that protects the thin filaments and does not allow the myosin heads to create a cross bridge. So we would refer to this as the sliding filament model, right? So in this process, calcium is going to be released. And what happens is the calcium binds to a complex that is covering the actin filaments. It's called the tropomyosin-troponin complex. And that exposes the actin filaments, allowing the myosin heads to make contact with them and allowing them to then pull and slide the filaments closer together, shortening the entire sarcomere. And they will stay in the contracted state as long as calcium is present. So in order to get the muscle back into a relaxed state, we have to use energy to pump the calcium away. Once the calcium is removed, then the sarcomeres elongate and go back to their pre-contraction lengths. All right, so here we see the actin filaments up here, and the myosin filaments are down here, and we see the heads are there. And the calcium is not present yet. However, when calcium binds to the tropomyosin complex that surrounds the actin filaments, it allows a certain area on the actin filament to become exposed, and then the myosin heads latch on, creating what's called a cross bridge. Here we see a cross bridge happening between the myosin filament and the actin filament. And this happens as a result of calcium being present. When this happens, we have the sliding of the two filaments, which is called the power stroke. And this is what's going to cause the whole sarcomere, the contractile unit, to shorten. Okay. As we want to go back into the relaxed state, we have to use energy to remove the calcium. Once we remove the calcium, the tropomyosin troponin complex slides back into a position that covers the actin filaments, not allowing the myosin heads to make a cross bridge. And that's going to cause the muscle cells to go back into a relaxed state. So it's interesting that ATP, the energy molecule that we need, right, is used not really for the contraction portion, but it's used to get the muscle to go back into a relaxed state. So this is all beginning at the neuromuscular junction. The neuromuscular junction is the tiny space that exists between the neurons, all right, so motor neurons and muscle cells. And if I want to move one of my muscles, I have to send a nerve signal that would begin in my brain and then travel down to the spinal cord. And it would go onto what's called a motor neuron. And this would leave the spinal cord and go down to the muscle cells. And what's going to happen is this is going to cause a muscle contraction. Let's take a closer look. So here we see the neuron, the motor neuron coming down, and this is the muscle cell here. There has to be an exchange of substances here, though, right? There's a chemical that is being released. If we look closer at the very end of the neuron, we could see there's a space. These two things don't touch each other. This neuron, this nerve cell, and this muscle cell are not touching. There is a space. We call this the synaptic cleft or the synaptic space. And a chemical needs to move across this space. This chemical comes in the form of what's called a neurotransmitter. The neurotransmitter that is released from motor neurons to move our skeletal muscles is going to be called acetylcholine. And there's receptors on the muscle cells. When the receptors that are specific for acetylcholine bind to the acetylcholine, it's going to cause a graded potential, and then eventually it's going to cause the beginning of a contraction cycle as enough of these are starting to bind. This contraction cycle will continue as long as this acetylcholine is binding to this. But there are enzymes in here that will break these things down immediately, and that's how we get the contraction to stop. There's a couple different types of muscle cells. We have fast twitch fibers. These are called glycolytic fibers. When you're doing push-ups, right, you're using your fast twitch fibers or glycolytic fibers because these fatigue very quickly. We can't do push-ups for 3 hours, for 2 hours, right? We could do it for maybe 2 minutes before we run out of gas, right? So they give us short bursts of energy and contractile power. They have fewer mitochondria, and it's these types of muscles that experience hypertrophy. So when we go to the gym and we work them out, they will actually grow. Okay. Slow twitch fibers are oxidative. They appear more red. They have a larger blood supply associated with them. They have more mitochondria, and they have the ability to store an oxygen-carrying protein called myoglobin. These are less fatiguing and more aerobic. So these are the muscle fibers that we're using when we are doing long distance running. You can run for an hour. You can run for 2 hours. Some of us could run for 3 hours. And it's because it's a different type of muscle fiber that is being used. You work these out every day. They're not going to hypertrophy. They're not going to grow, right? They're just going to become more efficient. And what I mean by more efficient is the blood vessels that are associated with delivering the blood cells to the area are going to work more efficiently, and you're going to be able to use them longer and longer the more you work them out. Okay? All right, so this concludes the lesson for the skeletal muscular system, the lecture. If you have any questions, don't hesitate to email. And stay tuned for the lab for muscular system.